Assalamualaikum and good day. Hi, we meet again to continue studying a new subtopic, which is subtopic 2.2. It is about structures and functions, plasma membrane and organelles. Without wasting time, let us study now. First of all students must know that they should be able to achieve these 4 learning objectives. The first learning objective is to show the structures of typical plant and animal cells and state the organelles present. The second learning objective is students should be able to identify the structures and state the functions of the following organelles, which are nucleus, rough endoplasmic reticulum, smooth endoplasmic reticulum, Golgi body, lysosome, ribosome, mitochondria, chloroplast, and centriole. The third learning objective is students should be able to show the structure of plasma membrane based on the fluid mosaic model. And the last learning objective is students should be able to explain the structure of plasma membrane and the functions of each of its components. So overall, this subtopic, we can see that they are all about the structures and functions of organelles and also the structures and functions of the plasma membrane. The first organelle that you are going to learn is nucleus. This is a three-dimensional diagram of a nucleus. There are three main functions of a nucleus. The first function is it contains genetic material of the cell, which is called DNA. It also acts as the center to control the cell activities and also the cell division. And the third function of a nucleus is it produces ribosomes and RNA. The second organelle is the endoplasmic reticulum. This is a three-dimensional diagram of an endoplasmic reticulum. This is a two-dimensional diagram of an endoplasmic reticulum. Endoplasmic reticulum is the largest of the internal membrane and it continues with the outer membrane of the nucleus. The rough endoplasmic reticulum is flattened in shape. It is attached by ribosomes on its outer surface and it continues from the nuclear envelope. Whereas the smooth endoplasmic reticulum is tubular in shape. It is laid of ribosomes and it is interconnected tubules. The function of the rough endoplasmic reticulum is to pack and transport proteins synthesized by ribosomes that attach to it. The function of smooth endoplasmic reticulum is as a site of lipid synthesis involved in carbohydrate metabolism, and it also detoxifies drugs and poisons. The third organelle is Golgi body. A Golgi body consists of flattened membranous sac which is called cisternae. This is the cisternae. The functions of a Golgi body are, first, to involve in modifying, packaging, sorting, and shipping proteins and lipids which receive from endoplasmic reticulum. Second, to produce lysosomes which contain hydrolytic enzymes. And the third function is to produce secretory vesicles. Next organelle is lysosome. Lysosomes are spherical in shape. They are single membrane-bounded sacs. They originate either from Golgi apparatus or directly from endoplasmic reticulum, and they contain hydrolytic enzyme. Two main functions of lysosomes are they involve in intracellular digestion of macromolecules by phagocytosis. And the second function is they involve in autophagy, which is digestion process of worn-out organelles in the cells. Next organelle is ribosome. Ribosomes are dense. There are small granules with diameter about 20 nanometer. Each ribosome is composed of two subunits, which are large subunit and small subunit. They can be found suspended freely in cytosol or attached to the endoplasmic reticulum or nuclear membrane. Their main function is at the site of protein synthesis. Next is mitochondria. Mitochondria is a plural word for mitochondrion. Mitochondria are large organelles. They have rod shape and are enclosed by double membranes which are called outer membrane and inner membrane. They are special because they are able to replicate freely as they have their own DNA. They are functioning as the site for generating energy in the form of ATP. Next organelle is chloroplast. A chloroplast is a biconvex disc. It is bounded by a double membrane which is called outer membrane and inner membrane. It is a semi-autonomous organelle. Its stroma contain many thylakoids, enzymes, DNA, starch granules, and ribosomes. Its thylakoid contain chlorophyll to trap light energy for photosynthesis. Granule is a stack of thylakoid. Chloroplast is functioning as a site of photosynthesis during light-dependent reaction and light-independent reaction. Next organelle is centriole. Centrioles are hollow cylinders. In 9+0 arrangement of microtubule, they are present in pairs and they are held together at right angles to each other, which means 90 degrees. The function of centriole is they help in organizing assembly of microtubule during the cell division. You have finished studying about the major organelles in a cell. Now let us study a new structure which is called plasma membrane. This is the plasma membrane. As you can see, the plasma membrane have two major components which are phospholipids and proteins, which create a unique physical environment. Based on the fluid mosaic model, the membranes are composed of two layers of phospholipids with the globular proteins embedded in the bilayer. So, the term fluid comes from the phospholipids and the proteins, which can move laterally or move side to side. And the term mosaic comes from the proteins which are embedded in the phospholipid bilayer, like the mosaic tiles embedded in the mortar. Plasma membrane is an amphipathic molecules, as they have both hydrophobic regions and hydrophilic regions. Plasma membrane have four major components which are phospholipid bilayer, two types of proteins which are the integral proteins, which partially or fully embedded in the phospholipid bilayer, and peripheral protein, which are attached to the outer and inner surface of the membrane. Cholesterol, which stabilize the plasma membrane structure by restricting the movement of the phospholipids during warm and cold temperature. And the last component is carbohydrates, which act as cell identity markers. Carbohydrate plus proteins becomes glycoproteins. This is the glycoproteins. Whereas carbohydrate plus lipids becomes glycolipids. This is the glycolipids. That's all. Good luck everyone. Until we meet again in the next lesson. Bye-bye.